Welcome dear students. I'm back again, myself Dr. Ajay Similti from HNB Garhwal University, Central University, Srinagar, Garhwal, Uttarakhand. So in the series of our lecture on novel system, today we are going to discuss the phytosomes and pharmacosomes. Apart from the synthetic drugs, it has been felt that the phytoconstituents can also be delivered in the form of NDDS. So it's an attempt. Phytosomes is an attempt in this context, apart from the synthetic drug delivery systems. So let's open up our discussion on the phytosomes and pharmacosomes. After completing this lecture, you will be able to define phytosomes or pharmacosomes, enumerate the advantage and limitations, distinguish it from liposomes, and enlist the preparation methods of phytosomes or pharmacosomes. We'll be covering this lecture in the following heads like definition of phytosomes and pharmacosome, advantages and limitations, their difference from liposomes, and methods of preparation. Now, novel delivery systems has been a major thrust area in the post-GATT era. And the Indian challenge lies in exploring the knowledge of Ayurveda and the medicinal plants. The herbal drugs still enjoy a major share in clinical practices. Bioactive components, phytoconstituents, when they are needed for oral delivery, they need the aqueous solubility for dissolving into the gastric fluid. They need the lipid solubility also for permission across the biological membrane and reaching the blood circulation. Remember that animation we saw in the last lecture, that drug needs to go into the solution in the form of dissolution process, then it is needed to pass across the biological membrane. So dissolution as well as the permeability both are needed. So solubility as well as the permeability both are needed for a successful oral drug delivery. Poor absorption and low bioavailability due to multiple ring molecular structure and lack of adequate lipophilicity or hydrophilicity may be solved by various means like developing the more soluble prodrug, preparing the more soluble complexes with complex agents like metals, cyclodextrin, phospholipids, lecithins, etc. So, what is the need of the era? We need to develop a potential NDDS for improved bioavailability and clinical efficacy. Phytosomes or pharmacosomes can be the answer of this problem. Phytoconstituents like flavones, xanthones, anthraquinones can be converted into the lipid compatible molecular complexes which are called phytosomes. Phytosomes are basically the deuterionic phosphatidylcholine complex of phytoconstituents which result into the better stability and bioavailability than the conventional herbal extract. So it's a method of modernizing the Ayurvedic drugs, or you can say herbal drugs or phytoconstituents. Phosphatidylcholine, which is a carrier, it is a natural carrier of the phytoconstituents for this kind of NDDS. Let's see the phytosomes. That intermolecular bonding of polyphenolic phytoconstituent with PC, phosphatidylcholine, proved to be specific and stable, and the resulting molecular complex is more soluble in lipophilic and organic solvent. When they are treated with water, they form micellar shape, forming structures which resemble the liposomes, but they are quite different from liposomes, which we are going to discuss in the coming slides. Phytosomes are reported to have 4 to 6 times more bioavailability than the conventional herbal extract or their parent drug. Okay, so they differ they look like liposomes, but they are not liposomes. Okay? We will be discussing liposomes also in our coming lectures. But let's see. In case of synthetic drugs, various common drugs like NSAIDs, like aspirin, diclofenac, aceclofenac are associated with low absorption due to poor water solubility and GI toxicity, gastrointestinal toxicity. Generally, the liposomes like lipid systems prepared so far are not stable and not so effective as to make the effective delivery of drugs clinically possible or feasible. Pharmacosomes as no drug system were first reviewed by Vazoglu in 1986. They are defined as colloidal dispersions of drugs bound to lipids and may exist as ultrafine vesicular, micellar or hexagonal aggregates depending on the chemical structure of the drug lipid complex. So what are the silent features? They have got the increased entrapment efficiency, easy removal of untrapped drug from the formulation, no loss of drug due to leakage, no problems of incorporation, and no influence of uncaptured volume and drug bilayer interaction on entrapment efficiency. We'll be discussing it in more detail later. Now let us define. We were telling that they look like liposome, but they differ. How they differ? Let's discuss. As far as the principle is concerned, liposome is on the basis of is in liposome incorporation of drug in aqueous or lipid phase of a mixture of lipids, where the physicochemical properties of the carrier and release of drug will be function of different lipid used. While in case of phytosome or pharmacosome, there is a electrostatic bonding of a drug to a lipid forming a complex, improving the bioavailability. The physicochemical properties depend on the drug as well as the lipid. So both, on both of the part they depend. As far as if you take the loss of drug, so in the loss of drugs, in liposome through leakage, loss of drugs may occur, while in the pharmacosome or phytosome there is no leakage since the drug is electrostatically bound, but loss of drug by hydrolysis is possible. Okay, hydrolysis may cause the loss of drug. Volume of inclusion in the liposomes, it is decisive incorporation of drug molecule, while it is irrelevant here since the drug is bound. If we talk about the surface charge, it is achieved through lipid combinations in liposomes, while it depends on the physicochemical structure of the drug lipid complexes in case of phytosomes and pharmacosomes. Membrane fluidity, if you are discussing that, it depends on lipid combination and presence of cholesterol. Fluidity influences the rate of drug release and physical stability of the system. While in case of phytosome or pharmacosomes, it depends on the phase transition temperature of the drug lipid complexes. There is no effect on drug release rate since the drug is bound there. Release of drug in case of liposomes, it occurs through diffusion through the bilayer, desorption from the surface, or release through degradation of the liposomes. While in case of phytosomes or pharmacosomes, it is by hydrolysis, including the enzymatic hydrolysis. In case of if you take the physical stability parameter, it is relatively good aggregation through the double valence cations may be there, while in case of phytosome or pharmacosomes, it depends on the physicochemical properties like drug lipid complexes. Overall, it has got the better stability than the liposomes. Now, however, we will be discussing the application part in quite detail in later section. But for the NSAIDs like aspirin, diclofenac, hormones, insulin, salmon, calcitonin, miscellaneous like pindolol, bipronol, acyclovir, taxol, propanol hydrochloride can be prepared in the form of pharmacosomes. Simply, if you simply say that if we are taking the phytoconstant, we say them phytosomes. If we take the synthetic drugs, pure synthetic drugs like NSAIDs, we call them pharmacosomes. Okay, phytosomes is rather a trademark basically by a company Indena. However, it has become generic to use it in case of phytoconstant and lipid complexes. So far, this figure we have taken from the Indena's silymarin complexes of phospholipid with the trade name of Silifos. It is basically silymarin is a mixture of sildenafil, silcristin and silabin. So it forms a good complex, phospholipid complex. The phytosomes have been prepared and used only for the cosmetic agent so far and dietary supplement, but phytosomes as herbal system have enormous potential in the enhancement of biopharmaceutical properties of phytoconstituents. We are developing phytosomes of selected bioactive phytoconstituents like flavones, xanthones, terpenes, anthraquinones, etc. from some medicinal plants. So merits, if we say the phytosomes show the following merits. What are the merits? It enhances the absorption of herbal constituents and hence the bioavailability. It enhances the solubility of bile to herbal constituent and facilitates the liver targeting in this way. And the absorption of chief phytoconstituent is improved in phytosomes or pharmacosomes. I can say its dose requirement is also reduced. So dosage requirement is also reduced, which is a peculiar feature of NDDS. Phosphatidylcholine used in preparation of phytosomes, it's a carrier, okay, but it also acts as a hepatoprotective. It's a hepatoprotective, hence it gives a synergistic effect. So carrier is also a natural one. So you can say it's a completely natural NDDS. Unlike liposomes, bonds are formed between the phosphatidylcholine molecule and phytoconstituent in phytosomes. So the phytosomes show a better stability profile over the liposomes. Now, what are the limitations of these PL, phospholipid complexes, phytosomes or pharmacosomes? The use of lipid complexes for improving the solubility may be limited due to the incompatibility with the phospholipids. Drug PL incompatibility may be there. The presence of PL may sometimes, that phospholipid may sometimes make the system more hygroscopic. Problem happens practically if you take, it is very hygroscopic. If the higher purity grades you will take of phospholipids are used, it will show hygroscopy. Secondly, all drugs cannot form the complexes with the phospholipids. Requisite criteria is the presence of free hydroxyl group for interaction with the phospholipid. This is utmost required criteria for drug, and any drug can't be complex with phospholipid. You will have to identify the drug which is having a free hydroxyl group for interaction, for available, it is available for interaction with the phospholipid. So then and only then the structure will be stabilized in the form of a complex. Now let us have a bird's overview on the commercial phytosome preparation. They are the Celebin phytosome. They are the trademark of Indiana, as I have told you. Ginkgo phytosome, Panax ginseng phytosome, green tea phytosome, super milk thistles extract, grapeseed phytosomes, hawthorn phytosomes. They are made from Celebin, Ginkgo biloba, flavone glycosides, ginsenosides, epigallocatechin, thiogalate, and Celebin procyanidolic oligomers, PCOs they are called, and flavones, and they are indicated in various things, especially where they food products, supplements, and hepatoprotective, and in some cases in cardiovascular diseases for hypertension, let's say in case of Hawthorne phytosomes. Let's discuss the properties of phytoconstituents which can be complex. As we discussed a little bit earlier, that any drug possessing an active hydrogen atom, like in free carboxylic acid group, free hydroxyl group, free amino group, it can be esterified to the lipids with or without spacer chain. Drug with very low water solubility or very low lipid solubility are the ideal candidate, provided they are having the free hydroxyl group available for interaction with the phospholipid. Okay, both of the things are required. Okay, now let's focus on the chemistry of phospholipids. Phospholipids are small lipid molecules in which the glycerol is bonded only to two fatty acids instead of three, as in triglyceride, with the remaining site occupied by a phosphate group. As you can see in this diagram, phosphatidylcholine is a polar lipid. It is present in commercial lecithin in concentration of 20 to 90%. So depending on your feasibility, in general, as it is my experience that the lipid 80% is far more suitable for laboratory purposes when you are developing the phytosomes, because the lower grades will have their own problem. They will, handling problem will be there, they will be greasy there, while if you go for the higher purity grade, they will be more hygroscopic, you can't handle them properly, and even in the characterization is very typical, because impurity pose several problems in various spectroscopic techniques. Okay, now as we have discussed that phosphatidylcholine is a natural carrier. It has got various therapeutic activities also, like it is effective hepatoprotective agent, it lowers the serum cholesterol levels, it protects against the liver cancer, and supplements of phosphatidylcholine may help in the treatment of Alzheimer's disease. So the polymer is completely natural and even it is useful. Okay, and it is used as a dietary supplement, so it's a complete natural novoactive system, and this is the beauty of this NDDS. Let's have a gross approach of synthesis, how we can prepare or synthesize the liposomes, or sorry, phytosomes. Any drug which is possessing a free hydroxyl group or free acid group, COH group. So when the drug containing the COH group or free hydroxyl group binds with the lipids OH groups, it forms the complex. If COH group is not present, but another active hydrogen atom is there, like hydroxyl group, free hydroxyl group, amino group, esterification is done by means of a spacer chain, spacer group, spacer arm technique. So what will you do, that you will use a spacer chain, and like spacer arm of succinic acid, you will add, and then the hydrophilic part and lipophilic part will be combined with the help of spacer arm chain, and they will form a stable amphiphilic product in the form of drug lipid complex. You can call it phytosome or pharmacosome depending on the drug used. Okay. Solvent evaporation, supercritical fluid, and anhydrous co-solvent difflation methods. These are the three major methods of preparation. Solvent removal, supercritical fluid, in which this main method is SAS, supercritical anti-solvent precipitation. It has got the two sub-techniques, gas anti-solvent method and solution enhanced dispersion by supercritical fluid. So these are the major techniques. We will discuss them one by one in our coming section. Now let's take the most widely used technique, that is solvent evaporation techniques. As far as myself is concerned, I have been using this technique, as well as the phytosome development I have been engaged in for last 12 years. So I have used particularly because it is very simple technique you can employ in your laboratories. What you are going to do in this method, phospholipids and the drug are taken into some molar ratio 1:1 or 1:2. They are dissolved in a common organic solvent like dichloromethane, tetrahydrofuran, methanol, etc. When you have dissolved them in a common solvent, what is the next step? The solvent is evaporated off under reduced pressure under vacuum. The drug is present. The drug, if for example for synthetic drugs which are in salt form, they have to be converted into the acid or base form for adding to the common solvent. Okay, so you will have to acidify those drugs so that they may have the free hydroxyl group available for interaction. Then the complex is obtained as the dried residues which are collected and placed in vacuum desiccators. So you can have these complexes, and depending on your requirement you may lyophilize them if it is feasible and if it is available with you. Now supercritical fluid method. To enhance the dissolution of pharmacosomes and to simplify the experimental procedures for preparing the complexes, supercritical anti-solvent precipitation is one of the SCF technique, supercritical fluid technique. It is becoming promising technique, to produce micronic and submicronic particles with controlled size and size distribution. The process is characterized by the very mild conditions of temperature, and smaller particles can be obtained depending on the drug and process condition when compared to the common industrial combination techniques like jet milling, liquid anti-solvent precipitation and crystallization. Particle size is particularly related to the dissolution of drugs and thus significantly affect their bioavailability. That's why it is very important, so the SCS process may increase the dissolution of pharmacosome. We are aiming for improving the biopharmaceutical characterization, biopharmaceutical properties like solubility, dissolution, okay, with the help of these NDDS. Two different technologies named gas anti-solvent, and solution enhanced dispersion by supercritical fluid SEDS belonging to SS process. We may use these techniques. However, they are specialized techniques. I'm not going into the detail. You can refer the further reading for these processes. However, these are not basically possible in our simple laboratories. Okay. In the anhydrous co-solvent lyophilization method, Basically, it's a lyophilization method. In this method, the drug and phospholipids are co-dissolved in a little, about 1 ml organic solvent like dimethyl sulfoxide and or glacial acetic acid with gentle agitation until the formation of a clear mixture. The resultant homogeneous solution, is then freeze dried overnight at a temperature of minus 40 degrees Celsius and under vacuum of 10 Pascal. The resultant complex is flushed with nitrogen and stored at 4 degrees Celsius. Okay, many scientists have prepared the insulin phospholipid complexes and other complexes with this method, lyophilization method. And lyophilization, you cannot take, again, rationality is the issue. You cannot make, you are not supposed to make any phytosome or pharmacosome with this method. The rationale must be there. Why people are preparing the insulin phospholipid complex with this method? Because insulin is unstable. Okay, that's why the lyophilization remains the choice only for salmon calcitonin maybe also. So, to conclude, as if in the preparation method, I would say that you must go with the solvent removal method, solvent evaporation method, and try to develop the phytosomes. I have developed lot many phytosomes as well as the pharmacosomes with published results from my laboratory, where my research team has done wonderful work on the phytosomes and pharmacosomes. Uh, and basically all the methods in each and every technique we have used the solvent removal method or solvent evaporation method successfully. Now, though these are the drug characterization methods of phytosomes and pharmacosomes, drug content, percent drug loading you must go, then SEM, TEM, FTIR, in among the spectroscopy, FTIR, NMR, any kind of NMR can be used depending on the drug. DSC, the thermal analysis, solid state characterization by the X-ray diffraction, XRPD, high resolution XRPD may be adopted for seeing the crystallinity, solubility, partition coefficient. Obviously, what change it is being brought by the NDDS can be seen by the solubility study or the partition coefficient studies or the permeability study. In vitro drug release study and in vivo activity studies are the characteristic parameter. However, we are going to discuss in detail the characterization in our coming lecture. We will be discussing these characterization because the characterization is very important part in this, and then we will be discussing maybe the application also one by one in our next lecture. These are some of our work, our references. You can go ahead with the further reading. You can have the further readings with these resources. Now, friends, let me summarize the whatever we have read, whatever we have discussed in our lecture. The phytosomes are the phytoconstants and phospholipid complex. When in the lipid complex, when the drug is phytoconstant, we call it phytosomes. If it is a synthetic drug, you can say non-plant source, we call it pharmacosomes. Remember that phytosomes is basically a generic word. It has been derived now, but actually the pharmacosome is the exact terminology, because phytosome is a trademark. Generally, we are not supposed to use the phytosomes because phytosome is the patented technology by Indena, and it's a trademark. It's a trademark. It's a trade name. Okay. So, however, because it has been the part of the various syllabi, so that's why we are calling it with the name of phytosomes. Pharmacosomes can also be developed for various problems of biopharmaceutical properties by low solubility, low permeability. So with the help of lipid complexes, we can improve the biopharmaceutical characterization, biopharmaceutical properties like solubility, dissolution and permeability. We can modulate these with the help of lipid complexes. Lipid complexes have got the various advantages over the liposomes and other techniques, other drug delivery systems. They are uh, providing the improved bioavailability and less leakage of drug because the drug is bound there, and they have got the better stability improved solubility, improved permeability, and thereby they are exerting better effect as compared to their parent drug. Low doses are required because more and more drug is being utilized by the system. So with these things, when we move to the methods of preparation, there are three major methods of preparation of phytosomes and pharmacosomes. We have also discussed its superiority, how it is differing from the liposomes, and it is very suitable, very important with respect to your exam point of view, because many times it is asked that how distinguish it, give the difference between the liposome and the phytosomes, or difference between the liposome and the pharmacosomes. Then you will have to give on the basis of various parameters. You can compare, which we have discussed, from how it is different from liposome to from how it is these phytosome and pharmacosome differ from liposomes. Okay. The solvent when we are coming to the method of preparation, just wrapping it up: solvent removal method, SCF supercritical fluid method, and lyophilization method. These are the three major methods of preparation. And as we have discussed, in the laboratory setup, the solvent removal method or solvent evaporation method is the most most convenient, simple, effective, reproducible technique which you can apply in your laboratories. Okay. uh, and it is I can recommend from my own experience, because we don't have the facility. However, if you are having the freeze dryer in your laboratory, you can go with the lyophilization techniques also, provided the rationale is good behind that. So far it's this much only on this topic, and we will be discussing the characterization and the application part of phytosomes and pharmacosomes in our coming lecture. Okay. So hope you have enjoyed this lecture, and we'll be coming back with the next part of this lecture in our next module. Thank you for being with us. Happy learning.